Welcome to the world of genetics. Today, we are going to talk about a new DNA sequencing method that is shedding light on the genome's so-called black box. This method, known as long-read sequencing, is able to read longer stretches of DNA than traditional methods, providing a more complete picture of the genome. One of the main advantages of long-read sequencing is its ability to accurately detect structural variations in the genome. These variations, such as deletions, duplications, and inversions, can have a significant impact on an individual's health. Traditional methods have had difficulty detecting these variations, but long-read sequencing is able to overcome this limitation. Another advantage of long-read sequencing is its ability to identify repeats in the genome. Repeats, which are segments of DNA that are repeated multiple times, can also have a significant impact on an individual's health. They can cause genetic disorders such as Huntington's disease and Fragile X syndrome. With long-read sequencing, these repeats can be identified with greater accuracy and at a higher resolution. Long-read sequencing is also helping to unlock the secrets of the genome's black box by providing a more detailed view of the genome's organization. It's allowing researchers to identify new regulatory elements and understand how they control gene expression. To conclude, long-read sequencing is a powerful tool that is shedding new light on the genome's black box. It's helping researchers to better understand the genetic basis of health and disease, and it's providing new insights into the genome's organization and function. With this new technology, the future of genetics is looking brighter than ever.